Good afternoon to all of you. We are delighted to have you with us today for the first one in 2022. To introduce today's speaker, Professor Dr. Marcel Utz of the Department of Chemistry at University of Southampton, UK. I would like to ask his host, Professor Dr. Jan Corving from the Institute of Microstructure Technology, IMT at KIT, who successfully nominated Professor Utz for an International Excellence Fellowship of KIT. Please, Professor Corving, the screen will be yours.
This is my pleasure to introduce today Professor Marcel Utz, who is Professor of Magnetic Resonance at the University of Southampton in the UK. Marcel's interests has a very interesting overlap with my own interests for magnetic resonance at the microscale. And this is really what has driven over the years our contact and our collaboration from the research point of view. Marcel is very prolific. He's authored more than 75 articles in major journals. He's written several book chapters. He started off with a bang by getting a medal from the ETH for his PhD thesis, the highest honor you can get at the ETH. And when he moved to the United States, he won an early career award by the US Science Foundation. And when he moved to the UK, he got a Marie Curie Career Integration Award within the 7th EU Research Framework. Marcel, we very much look forward to your talk.
Thank you, Jan, for this very generous introduction. Now I have to live up to the expectations you've generated. I'm not sure I'll be able to do this. So thank you very much for giving me the opportunity to speak to you tonight. Thank you for being here. I would also like to thank the International Office of KIT for making this possible. It's a great honor, pleasure indeed, to stay here in Karlsruhe for the last few weeks. I've been here last summer for a few weeks as well, together with my family at the time, and we've all had a wonderful time, both personally but also scientifically. Because I tend to forget, I would like to start off with just flashing up a brief acknowledgement to many collaborators who have contributed to the work that I'll be presenting today in various ways. I will say more about the specific contributions at the end of the talk, but I'd like to use this opportunity to point out that no man is an island. The science, like any cultural activity, is a collective phenomenon, and I'm all the more grateful for this opportunity to interact with people in person here. This is not something to be taken for granted, particularly not in our coronavirus infested times. Maybe a quick word about where I'm from. So magnetic resonance has a long history and a proud tradition at Southampton, and it's a focus area at the university and specifically within the School of Chemistry. There's a section consisting of 7 principal investigators, including myself, who work on various aspects of magnetic resonance methodology, but also applications. We have one of the larger NMR centers in the UK, and we're also, of course, actively involved in developing hardware and novel methods, some of which I will go into in the course of the talk. So let's back up a little bit. If you look at the grand challenges of our time, obviously, depending on who you ask, you will get slightly different answers to what they are. But I think we would all agree that the things I have listed here are indeed things that we have to solve, things we have to work on in our generation in the next maybe 25 years. And if you examine them a little bit more closely, then you realize that they all have something in common. They all revolve in one way or another around biology, because the big overarching question is the interaction between human society and the natural world. And in order to make any progress or solve any problems in that space, you require insight into the biology, and that insight should be quantitative. So you have to look at biological systems and derive quantitative information from them in order to make any progress. What do I mean by a biological system? Well, I mean literally almost everything. A system can be an ecosystem like the Great Barrier Reef, or it can be an individual animal like this mouse, or it could be an organ inside of that animal, or of course inside of a human being, or it could be a type of cell or an individual cell inside of that organ fulfilling a function in there, or indeed a organelle inside of that cell. Or finally, it could be a molecule or a collection of molecules that play a role in the function and in the structure of such a system. So what do these biological systems have in common? There are some aspects that they all share, even though they're very different in scale, in size, in importance, but they all share that they're part of an environment. They interact with their surroundings. Even the biggest one, the barrier reef, interacts with the currents of the ocean, with indeed the human world in various ways, etc. And the same is true at every scale that I've mentioned. The next characteristic that I'd like to point out is that each and every one of these systems, right down to the lowest one here, right down to the right, where we look at individual biomolecules, is hugely complex. If in case you're wondering, this is a ribosome, the crystal structure of a ribosome. So complexity is a running theme in biological systems. And finally, these systems tend to be diverse. So if you have figured out how to deal with a particular system, well, that only gets you this far because there are many different individuals of that system, which are all subtly different, and you may have to deal with that diversity in specific ways. Now, quantitative information plays an enormous role in biology nowadays, and the role is only ever increasing. There is a hierarchy one can employ here, starting from the genetic information, going through the transcriptome, which of course depends on the genome, which then leads to the proteins that make up the function and structure of most biological systems. And then of course, the fuel that runs the entire machinery is the metabolism. And of course, metabolism is driven by proteins, their expression and their interaction with small molecules. So this entire chain here drives what we call systems biology. And there are many important outcomes that are supported by this kind of systems biology, systemic research. I've listed just a few examples here. Now, specifically, what I'd like to talk about today are lab-on-a-chip systems. These are systems that are microfabricated structures that are platforms for experimentation with biological systems. And they specifically deal with each of these three different characteristics that I've mentioned before. So, for example, the fact that every biological system is part of an environment that it interacts with can be mapped into very specific structures in which the environment can be controlled. So here in this example, the lung cells, endothelial and epithelial lung cells, are grown on a stretchable membrane, which can then mimic the function of the lung. The complexity that is inherent to almost all biological systems is reflected in data-rich analytical techniques that are being used. So for example, here, this is a perfusion reactor where hepatocytes are grown, so primary liver cells are grown. And in this particular study, the authors used the expression levels of various biomarkers to study the differentiation and the viability of these cells. And then finally, the diversity that comes with biology also can be accommodated in lab-on-a-chip systems. And here I picked an example from Ryan Kelly's group at Brigham Young University. The idea here is that a microscope is equipped with a strong laser, which can be focused on a particular point in the image. That point then gets destroyed, the cells in it get vaporized, and the vapor gets picked up by a little droplet of a chip that sits on top of the microscope slide, and then an automated mass spectrometer picks up that droplet, and from that, the proteome of that particular tissue in that particular site in the tissue can be analyzed. So now let's back up a step and talk about magnets for a moment, and then we'll bring the two things together, microfluidics and magnetic systems. So you all are familiar with magnets. Of course, I'm aware that some of you in the audience are experts in magnetic resonance, so you may lean back now, but there are many of you who are not, so allow me to indulge a little bit in talking about the fundamentals here. So you all know this device. It's a compass needle here that tends to point north, and it's used for orientation. And we all know that magnets have something to do with rotation. This can be exemplified, for example, here on the top right in the Einstein-de Haas effect. So here is a steel rod, the magnetic moment of which is periodically inverted, and it leads to the rod starting to rotate. So there's something actually about rotation and magnets. And indeed, the magnetic field of the Earth is, of course, driven by the Earth's rotation, together with convective phenomena inside of the Earth's molten core. So magnets, like a compass needle, are typically magnetic dipoles. That magnetic dipole in the compass needle couples to the external magnetic field generated by the Earth. And as you all know, I can make use of this. And everywhere I take the compass, it always points towards a point that's near the geographic North Pole of the Earth. How about atomic nuclei? It turns out that they are also dipole magnets. Each atomic nucleus carries a magnetic moment, and that is then the basis for nuclear magnetic resonance. So the way this works is that each of the protons, in particular here on this molecule, a molecule of lactic acid, so each of the protons here carries one of these little dipoles. And if I have a solution of lactic acid in water, say, then of course all these little dipoles are swimming around in that solution, and they tend to point in random directions as long as there is no guiding field. If I switch on a guiding field, then something not totally unlike the interaction between the compass and the Earth's magnetic field happens. These dipoles tend to align with that external field. Now, there is a counteracting force, or a counteracting principle, and maybe it's a better word, and that's thermal motion. And that ensures that mostly the system stays disordered, but there is a slight bias of pointing north as opposed to pointing in another direction, so pointing along the external field. And that little bias is what is the basis of nuclear magnetic resonance. Now, I should tell you that atomic nuclei not only carry a magnetic dipole moment, they also carry a property that's akin to angular momentum and has something to do with rotation, or behaves like rotation from the point of view of quantum mechanics. And indeed, the two phenomena, the magnetic dipole moment and that rotation, or that angular momentum is more precisely, are locked together. They are flip sides, if you will, from one and the same coin. And actually that lock is absolute, so you cannot decouple those two from one another. And those two together now give rise to an interesting phenomenon. When you expose them to an external magnetic field, then these spins tend to start to precess. And that precession, that motion around this precession axis here, happens with a frequency that's characteristic of the type of nucleus and proportional to the external magnetic field. And it depends in subtle ways on the precise environment of that nucleus. And that makes these nuclei, of course, the ideal spies that tell us something about the system. Since this precession is a reorientation of a magnetic moment, we can actually pick up the electromagnetic radiation that it generates with a coil or some other structure. That results in a time signal like this, in an oscillation, which we can then Fourier transform or frequency analyze, and we end up with a spectrum like this one here. So this is an NMR spectrum. Look at the scale here is in parts per million, and that means the scale here from right to left, by the way, by peculiarity of history, this is common in NMR spectroscopy. So this indicates a very small fraction out of the total spectrum, because all of the protons in this molecule resonate or precess at almost exactly the same frequency, but there are subtle differences. And that gives rise to these different signals. Some of these are not of interest. There is a calibration compound in here called TSP, and one signal here is due to water in the solution, but the other two signals are due to the lactic acid, and specifically due to the protons I have marked here with dipoles. And there are two that are chemically, two classes that are chemically different from one another. So there is a methyl group here, which contains 3 protons that gives rise to this big signal, and then there is a lone proton here on the a bit further to the right, and that gives rise to this signal here. So now, if I have a mixture from a metabolically active biological system, then I can see literally hundreds of metabolites in the NMR spectrum of that. And you will recognize here, by the way, this is brain from brain tissue, and you recognize already the signal here of lactate, which I've just shown you. But now there are many, many others that are superimposed here in this spectrum. And indeed, we get systemic information on the entire metabolite soup, the entire metabolome of the system from this spectrum. This information is generic because every metabolite that I can think of contains protons, and no 2 metabolites will produce exactly the same NMR spectrum. It's quantitative because you can just integrate these peaks and thereby determine the concentration of the different compounds. And it's a non-invasive technique. The type of radiation, the frequencies involved are compatible with life, and many of you may have had the somewhat dubious pleasure of a ride in an MRI machine that works by almost exactly the same principle. And that's, of course, living proof that NMR is a biocompatible, non-invasive technique. It doesn't destroy biological samples. Now, modern standard NMR systems look like this. They're actually rather big. This is about 3 meters tall here. They consist of a large magnet and some electronics in this box here. And the samples are usually about 200 to 600 microliters. They go into these types of little tubes here. And inside of this magnet is a vertical bore here that contains some electronics and some ancillary hardware. And the sample is then lowered into the NMR magnet here, in this case by an automated system, a sample lift that puts these little tubes here into the middle, lowers them into the bore where they sit inside of this structure here. You can sort of see that peek out from the bottom here with all the cables attached. And we call this a probe. That is essentially a system to carry radio frequency signals to the sample and the time signal that I've shown you earlier back from the sample for analysis. So because so there's a number of reasons, but the small amount of thermal order that I mentioned earlier that is caused by the external magnetic field is one of them. NMR is not a very sensitive technique, and that shows up in the amount of sample that is typically used in standard systems, almost a milliliter. That's a significant amount, much more than is used for other spectroscopic techniques, that's for sure. By the way, I would be remiss in not mentioning that Karlsruhe plays a very important role, continues to play a very important role in the field of magnetic resonance, both academically, of course, but also industrially. And there are pioneers that have really contributed to this field that have their roots here that I should mention. Heinrich Hertz, of course, is absolutely crucial, was absolutely crucial in, we wouldn't be dealing with electromagnetic signals the way we are without his contributions. And the other person I would like to mention is Professor Günther Laukien, who is one of the co-founders of Brucker, which of course are in Rheinstetten, just outside of town. So now, microfluidics and NMR, how can we put these two together? Now, obviously, like I've shown you before, lab-on-a-chip microfluidic systems are designed to mimic or to experiment with a biological system in exchange, in contact with an environment. And both are represented in some kind of microfluidic perfusion device. These devices are typically run or operated in almost the same recurring scheme. First, you have to seed the microfluidic device with the biological system, be it cells or a tissue slice or even a small animal. And then over time, you will culture it by perfusing it with growing fluid, you give it nutrients, you control the temperature so that it finds the right environment to grow. And during that time, you may add some kind of stimuli. For example, you may expose it to a drug or an infectious agent or some other influence that you want to study. And at the end, after, say, a day, a week, or maybe a month of culture, you destroy the system and you analyze, for example, which proteins were made, which genes were transcribed, which genetic characteristics were inherited, if the system proliferated, etc. So there's a destructive analysis at the end. What we hope to do with NMR is complement this by giving information on the metabolic history of the system and then correlate that with the information that one would have gotten conventionally. So let me show you some examples in the remainder of the talk of how we go about this in Southampton, and actually some of the work I will show is in collaboration with Jan Corving's group as part of the Future and Emerging Technologies program that he had mentioned. So here is a very simple microfluidic device. It really consists of just a little sample chamber here with some access channels, and the holes you see here at the top are just for mechanical purposes to hold this structure together. I'll show you more closely how this works later. So here we have, to start with, instead of 600 microliters, we only have 2.5. So we start with very much less volume to look at. And what we've done here is we've taken the same kind of cell, MCF7 cells, which are rather easy to grow cell line. And we've grown them in two different ways. In the left side, of the left column, we've coated the inside of the chip with a substance that makes it difficult for the cells to adhere to the surface. So they have to adhere to each other, and they create a little blob like this. This is also known as a cell spheroid. Whereas on the right-hand side, we've done what is conventionally done. We've made sure that the chip is coated with proteins that make it easy for the cells to attach. So the cells grow in this 2D, more conventional spread out fashion. And what we were interested in is, can we distinguish the metabolism of these cells given these two different environmental influences? So we built a system that can take NMR spectra from this small volume and produce them with good enough sensitivity for analysis. And you can see here, this is an example of how the NMR system reflecting, the NMR spectrum reflecting the metabolic makeup of the solution surrounding the cells changes over time. So you can immediately see, for example, if the cells are making lactate, they're producing alanine, they're consuming glucose, the glucose signals become weaker over time, etc. So there's various differences we can see that are happening with these cells. And then at the end, after 48 hours or so of letting this experiment run, we could still use fluorescent analysis, which is destructive, to assess whether or not the cells were still alive. And green here indicates that they are alive. And you see this in the case of the spheroids, but also in the case of the adherent culture. So from this, we can now quantitatively interpret the spectra. The way we do this is by a very simple mathematical procedure. So you have the experimental spectrum shown in blue here, and the fit here in red is just the best superposition of the spectra of the individual compounds that we are interested in that we could get. So you essentially just add some alanine, some glutaric acid, some lactic acid, etc., together until the superposition spectrum resembles most closely the measured spectrum. From this, we can, because we do this as a function of time, we can then determine the metabolic rates in the spheroid and in the monolayer culture, and we can compare them. And the first thing we found was that the monolayer culture grows or metabolizes things much more quickly than the spheroids. But that's not the only difference. You see that, for example, in the case of alanine, excuse me, and glutamine, it's the sign of the metabolic rate that's opposite. So while the spheroids actually consume alanine, the cells in the monolayer culture seem to produce it, etc. Since we know from simple microscopic counting how many cells we have, we can then determine the metabolic rates per cell. And I should add that the number of cells we used for this experiment is relatively small. There are a few hundred or a few thousand cells, so 2,500 cells in that small 2.5 microliter volume. So what are the challenges in pulling something like this off? A big challenge is sensitivity. So I've already said this before, NMR is not a very sensitive technique compared, for example, to mass spectrometry. So you have to be very careful in how you design that pickup coil so that you're not losing any sensitivity and not letting in any noise. Another somewhat related problem is spectral resolution. If you make small structures that have nooks and crannies and corners in them, you tend to compromise the homogeneity of the magnetic field that broadens the spectral lines, and it affects spectral resolution. And then finally, obviously, we didn't build an NMR spectrometer from scratch in order to look at our microfluidic device. We had to somehow integrate a commercial system with a microfluidic device in order to do these experiments. So there's a systems integration challenge here that has to be addressed. For the sensitivity, we've worked quite closely on finite element modeling to come up with this sort of detector structure. So this is in lieu of the coil I've shown you earlier in the principal sketch, just a solenoidal coil. We're not using a solenoid, we're using this kind of strip line structure, and we have carefully optimized its geometry along with the geometry of the sample chamber in order to get the best sensitivity possible. We have then built this device into a NMR probe. I've shown you some pictures of NMR probes before, which can fit into a standard Brucker NMR spectrometer. And the only difference is that instead of the round cylindrical sample tubes, it accommodates a microfluidic chip shown here. And that allowed us to obtain spectra like this one that are really indicative of a large range of metabolites. And we can then get quantitative information from a much smaller volume than was conventionally the case. So let me shift gears and talk a little bit about precision cut liver tissue slices. So this is the core of the project that Jan had mentioned, the EU project, we call it Tissue MR, which has just come to its conclusion. The basic steps to get these tissue slices are shown here. This is a model that has been developed by our collaborators at the University of Groningen. So the liver, and this is a mouse liver here, is cut into cylindrical sections, which are then sliced using a specialized microtome, and you end up with these 200 micron thick and about 3 to 5 millimeter diameter slices of intact liver tissue, which can be cultured either conventionally in well plates or in a microfluidic perfusion system. And the goal of the Tissue MR project, and that has kept us busy over the last 5 years indeed, was to come up with a microfluidic perfusion device that allows to keep these tissue slices alive and to analyze their metabolism in real time by NMR spectroscopy. Of course, that sounds like a cool idea, and it certainly did when we wrote the proposal, and we painfully learned that it's not as easy as it sounds. So we had over time to come up with a rather complex system consisting of many parts that provides the right conditions, the right amount of oxygen, the right temperature, the right temperature stability, etc., to this tissue slice inside of a microfluidic chip, so that we can observe its metabolism over extended periods of time. And by extended, I mean 24, 48 hours. And we hope to continue to develop the system to go up to maybe 72 hours or maybe 1 week in the extreme. So by continuously monitoring these slices, we can get entire ranges of NMR spectra. So here we've taken a spectrum every few minutes over 42 hours. And in this specific experiment here that I'm showing you, we were interested in what happens to the metabolism if we suddenly starve the slice from oxygen. So at some point here, we stopped the supply of oxygen, replaced the carbogen, which we used before, with argon, which of course does not provide any oxygen to the tissue anymore. And you see that immediately there are changes in the NMR spectra. Now, some of these are indicative of pH changes. So that, of course, directly shows the acidosis, which is a result of lack of oxygen in tissues, is well known. But there are more subtle changes in here as well. And we have now the quantitative data to analyze them. Now, by the same kind of analysis that I've shown you before, we can disentangle the different metabolites from these spectra, and we therefore now get a wealth of information. We get metabolic concentration profiles over 72 hours or 48 hours, here it's 24 hours, of the entire perfusion experiment, and we can then start to look for trends. So in this particular case, for example, you see that the slice produced acetic acid. That's common for liver tissue to do. It also produced lactic acid here. You see that very clearly. And then there are some more subtle signs of metabolic activity. So for example, here's leucine. There is also some residual activity here in valine, L-tyrosine, which is produced from phenylalanine, etc. So we can actually get these metabolic rates, and we get quantitative information. You can do rigorous error analysis with these data. So you end up with metabolic rates in millimolar per slice per hour. And look at the numbers here, right? So 100 micromolar per hour per slice. That's not a very large consumption rate. We actually get reasonably small error bars here, and we can really see how this slice lost slowly its viability, as exemplified by the loss in actic acetic acid production rate. Now, what are we going to do with this? Well, one proof of principle experiment we have done is to compare tissue slices that have been primed with a drug that that's known to induce a state of the liver that is characterized by inhibited expression of or excretion of bile. So this is called cholestasis. It's a rather serious liver condition, and it's actually the reason why quite a number of drug candidates never make it to the market, because they have that as a side effect, and it cannot be tolerated. One important drug here is chloropromazine. This is an antipsychotic drug that is used, but it's known to have this side effect. And so we looked at the metabolism of tissue slices treated with chloropromazine and compared it to those that were not treated. And you can see that there are significant differences in the metabolic rates associated with a whole number of different metabolites, while others seem less affected. Now, just to mention, I briefly mentioned magnetic resonance imaging earlier. Now, the same structure, the same microfluidic device and NMR probe system can also obtain magnetic resonance images. And here I'm showing an example. And so this is the tissue slice here, so or at least a part of it, about the right half of a tissue slice. The NMR detector is only sensitive in a strip, and that's why you get this kind of strip-shaped image. But what I find quite remarkable is that one can actually see the histological structure of the liver tissue in these MR images, and we didn't expect the resolution to be this good, and we're quite excited when we saw this. We are now using this to study how quickly the tissue metabolizes contrast agents and get some information on tissue viability that way. Another example I'd like to briefly talk about, if I still have a moment, is automated titrations, which you can do in a microfluidic system. So here we mix two different fluids in the microfluidic device, and we observe the NMR response, or the NMR spectrum, as a function of the mixing ratio. This is called a titration. It's the probably first kind of experiment you may have done in your studies in your first year chemistry practicals, except we want to do it with NMR, and we want to do it in an automated way. So we built a microfluidic device that has valves in it, which we can pneumatically activate, and we can use them to peristaltically circulate fluid inside the chip, inside the NMR spectrometer. We use this to study a protein that is implicated in a very large number of signaling processes. Specifically, my collaborators in biology are interested in its importance to T-cell stimulation, so in its role in the immune system. Its protein, FIN, is a tyrosine kinase, and it is regulated by several docking domains, in particular this SH3 domain here, which actually happens to be a very stable protein, which makes it ideal to do NMR studies on. And what we did is we presented it with a peptide which is known to bind to it. That peptide plays a role in activating this kinase in cascade to T-cell activation. And we wanted to determine in a fully automated way the binding constant of that peptide with the SH3 domain. Now, experiments of this type are very commonly done in the study of drug candidates, in drug discovery, but also in diagnostics and in many other aspects of biology and the life sciences. The problem doing these with NMR is that you need a large amount of sample conventionally, because you end up making many samples at different concentrations. Now, with our system, we can do this in an automated way with a very small total amount of solution and a very small total amount of protein, less than 1/4 milligram, the entire binding study. And what we observe as a function of the mixing ratio is that the peaks in this two-dimensional NMR spectrum shift, and by correlating the peak shift with the concentration of the ratio between the peptide and the protein, we can determine quantitatively the binding constant. Now, finally, I very briefly mention, I'll just flash this up because I'm out of time, that we are not just simply resigning ourselves to living with the amount of polarization of spin order that is given to us by thermal equilibrium, but there are tricks in NMR that are very actively researched that allow ordering the spins sort of artificially in order to boost the NMR signal. So this active area of research, I think, very strongly benefits from the integration of microfluidics with NMR, which also in turn, NMR then can actually enable applications for which the sensitivity would otherwise not be good enough. So I don't want to bore you with the details here, but you'll by now recognize these microfluidic devices, and we can run chemical reactions on them that use one of the spin isomers of molecular hydrogen in order to create highly spin-ordered products which produce enormously enhanced NMR signals. And we've recently managed to do something similar, rather than with an artificial molecule, with an actual metabolite in an aqueous solution. And we're very hopeful that in the near future we'll be able to use this in order to study the uptake of fumarate in cells and tissue slices. So let me come to a couple of conclusions. I hope I've convinced you that NMR and lab-on-a-chip devices offer enormous synergies to enable and enhance biological research. Maybe you've also realized throughout the talk that this is a very interdisciplinary field. In my group, we are jacks of all trades. We do everything from finite element simulations of electromagnetic fields to dealing with the adhesion of cells and analyzing metabolic signals, etc., everything in between. So this is a highly interdisciplinary type of research, and the environment that KIT in particular have built in the last 10 years is an ideal fertile ground for this type of research. For the future, I see a few really raised big areas of hope. One is that manufacturing has made enormous advances recently. Additive manufacturing, but also, of course, microfabrication. And the reason this is so significant is that not all materials are compatible with NMR. The choice of possible processes and materials used to be extremely restricted because of the presence of this very large magnetic field. And that design space has enormously broadened as a result of the processes of the advances in particularly additive manufacturing. Another one is automation. I think there are great possibilities in integrating microfluidics and NMR in automated systems. An example, a very primitive, simple example I've shown you just now. And then finally, because we are extracting a very rich wealth of data from a biological system, analyzing and working with this data and using some of the data to drive the automation of the experiment, I think also holds great opportunities for the future. So finally, I would like to thank you for holding on for this long and staying with us. I would also like to thank my team at Southampton, shown here in a pre-COVID picture. So most of these people are still with us. Some of them have graduated. I would like to mention in particular Manendra Sharma, who's built the NMR probe that underlies most of our work. I'd also like to point out Sylvia Ostrovska, who has worked on hyperpolarization and is really an extremely productive student. I'd also like to point out, of course, my colleagues, Malcolm Levitt and Jörn Werner in Southampton. James Eels is a postdoc with whom we've collaborated extensively and still do. And finally, last but not least at all, of course, I'd like to mention my collaborators on the EU project that I've mentioned several times, Professor Verpoort at the University of Groningen, and my gracious host, Professor Corving at KIT. And I'd like to thank the international office at KIT for giving me the opportunity to come here as an International Excellence Fellow and to speak to you tonight. So thank you very much, and I'm very happy to answer your questions.
Thank you, Professor Utz, for this very interesting presentation. So now I would like to open the question session. Akira Miyazaki, one of our International Excellence Fellows who was at KIT in October and November. He's from the University in Uppsala in Sweden, so joined us also today.
There was like a superposition of different spectrum to, yes, that I was wondering it because it looked like a scalar sum, isn't it? I was wondering why it's not a vector sum. Aren't there any phase shift in the different spectrums which makes an interference to just distort the overall spectrum?
No, it's actually, that's one of the big advantages of NMR. So the presence of one compound in general does not alter the spectrum obtained from another. So you really just get a linear superposition. That has to do with the fact that, you know, in a physicist's parlance, that we are working with very weak coupled systems. So the response is very, very linear. And that's one of the advantages over, say, mass spectrometry, where if you have a compound, it will give you a signal. But if you have some other compound present at the same time, that may interfere with the fragmentation of the compounds that you want to study, and you end up with crosstalk. So we don't have that kind of crosstalk, and that's actually a crucial point.
Thank you.
I have two short questions, technically also. A, why did you use 80% oxygen? I would have used 20% oxygen because it sounds not natural. And B, you have this sawtooth figure in the acetic acid that the rate changes as a function of time. I say, okay, but why the sawtooth? This is a super high nonlinearity.
First question is because our colleagues in Groningen, from whom we have inherited this type of biological model, have found out independently before we got involved that growing these tissue slices in 80% oxygen environments keeps them alive, differentiated, and functional for longer, which is why they have fixed the protocol that they use in well-plate cultures to this value. And we essentially took this over and replicated it in the microfluidic systems. Now, that said, we are currently following up on this research and using the NMR data to optimize those conditions. And as you say, it isn't natural, and it may well be that the outcome of that research will be that maybe 80% oxygen is too much and we should reduce the oxygen partial pressure in the system. The second question is the sawtooth pattern. Yeah. And of course, that's because you're puzzled because I forgot to mention something. So what we do actually is we solve a problem. There is a contradiction, if you will, in this system between the need to supply large amounts of fluid so that the system remains oxygenated, with doing it slowly enough so the concentration can change in a way that you can measure, right? So those two don't necessarily go together. So what we do instead is we have a system that we pump both ways. So the liquid oscillates left and right across the tissue slice for a certain period of time. And then when the concentrations have changed by too much, or after a regular amount of time, we push that plug out and we replace it with fresh medium, and then we do the same thing again. And that results in this sawtooth pattern. I've looked at so many of these over the last couple of years that I forgot to mention it. Apologies.
It looks like stick-slip motion in mechanics. Thanks.
Thank you, Manfred.
Thank you once again, Professor Utz. You've really mentioned that your project is very interdisciplinary. That is also one of the main aims of our project, of International Excellence Grants, and also of this series. We would like to bring together the scientists from different fields and to exchange your ideas. I would like to say, everybody, thank you very much for your participation today. So I wish you a very good evening, and I hope that we'll see you at some of our other events, maybe also in presence.